When we talk about medical challenges, really tough ones,
Right.
I mean, figuring out how to treat the brain and the nervous system, it's got to be right up there.
Oh, absolutely.
Like, imagine you're trying to repair the most complex wiring imaginable, but all the little parts, they're just breaking down or not firing right.
That's exactly it. And for the longest time, we've had so few options, really effective ways to intervene and actually get therapies to where they need to be in the central nervous system. It's a tough nut to crack.
But for a while now, we've been hearing all this buzz about mesenchymal stem cells, or MSCs. They seem like, wow, this is it, a real breakthrough.
Yeah, they did. We were seeing, encouraging results in preclinical studies, animal models of all sorts of neurological problems, and it was all looking good after MSC transplantation.
Okay, so let's break this down a little. We transplant these MSCs, and we think, great, they're going to become new neurons and fix everything.
Right, that was the initial thought.
But then, here's where things get really fascinating. It turned out the more we dug into it, it wasn't necessarily the cells themselves, sticking around and transforming into new neurons. It was more about what they were releasing.
Exactly. The focus has really shifted, across the scientific community towards these tiny packages of information. It's what the MSCs secrete. We call them extracellular vesicles, or EVs.
EVs.
Basically, they're nanovesicles, think tiny sacs, packed with proteins, lipids, and nucleic acids. And these little guys seem to be the real heroes, the ones doing the heavy lifting behind those therapeutic effects.
So we're talking about using these little messenger bags instead of the whole cell. Fascinating.
Yeah.
And that leads us right to the heart of our deep dive today. Getting these MSC-derived EVs straight to the brain, and we're doing it by going through the nose intranasal administration.
It's a really exciting approach. These EVs, they're orders of magnitude smaller than the original MSCs. So they have a much better shot at getting through that blood-brain barrier. It's notoriously selective. Plus, their safety profile looks way better compared to transplanting whole cells. Potentially lower risk of triggering any unwanted immune responses.
And going through the nose, come on, that's got to be so much more appealing for patients. No needles, no surgery, just a sniff.
You got it. Intranasal administration, it's completely non-invasive, and it could be a real game changer in terms of bypassing the blood-brain barrier, potentially delivering those EVs right where they need to be in the central nervous system.
So imagine these EVs as these miniature delivery trucks. What kind of cargo are they carrying, and what kind of impact can that have on, damaged brain areas?
Think of a really diverse payload. They've got molecules that can put the brakes on inflammation, signals that can actually encourage new nerve cell growth, protective stuff that shields existing neurons from damage, and even enzymes that can break down those problematic protein buildups we see in so many neurological diseases.
So for this deep dive, we're setting out to understand how these intranasally delivered MSC-derived EVs actually get to the CNS, how they find their target, the injury site or the inflammation, and what kind of functional improvements we're seeing in the animal studies. Sounds like a plan.
Absolutely. Let's start by looking at why these EVs might actually be better than using the MSCs themselves.
Right. We touched on it earlier, but what were some of the limitations of just putting those MSCs directly into the body?
Well, research has shown that when you inject MSCs intravenously, surprisingly few actually make it to the injury site in the brain. A lot of them get stuck in the lung capillaries, those tiny blood vessels, and then the body just clears them out pretty quickly. So they're not really hanging around where we need them most.
So it's a delivery issue. They're not getting to the right address, at least not in enough numbers.
Exactly. Now, if we look at EVs, it's a totally different story. Studies show that these tiny vesicles, they can actually cross the blood-brain barrier and migrate to different brain cell types. And this is something their larger, parent cells, the bone marrow-derived MSCs, struggle with.
That's a huge difference in terms of getting the therapy where it needs to go. Now, what about the body's reaction? Were there concerns about immune responses with the MSCs?
Definitely. MSCs are living cells, so they could potentially activate both the innate and the adaptive immune system. That could lead to graft rejection in some cases. There were also worries about them causing clots, embolic events. And in some very specific lab settings, there were even concerns about them maybe promoting tumor growth or changing genetically after being in culture for too long.
Sounds like some serious potential downsides. How do EVs compare in terms of the immune system?
Generally, EVs seem a lot more stable and way less likely to cause a big immune reaction. Studies haven't found major histocompatibility complex, MHC class I or R molecules on their surface. Think of MHC molecules like, hey, this is me, flags on cells. EVs don't really have these flags, so they don't trigger that foreign invader alarm in the immune system. What's interesting is there haven't even been reports of them causing immune reactions in preclinical studies where they were given to different species.
Okay, that's a big plus for safety and maybe being able to use them more widely. But you mentioned that EVs aren't totally risk-free.
Right, they can't form tumors themselves because they're not cells. But there's a theoretical possibility that they could indirectly help pre-existing tumor cells grow or even cause clotting in some situations. These are definitely areas that need more research to make sure we understand and can manage any potential side effects.
So we need more data on long-term safety. But it sounds like for reaching the brain and avoiding immune problems, EVs have a clear advantage over using the whole MSCs. And you said their therapeutic effects can be just as good, if not better.
That's what the research is showing, yeah. Several studies have found EVs to be just as effective as, and sometimes even more so, than the MSCs they came from. Of course, comparing results across different studies is tricky. There are variations in dosages, the types of EVs used, size, what they're carrying, how they spread in the body, and even other stuff secreted by MSCs that might be playing a role. But using EVs instead of MSCs is a really exciting direction for treating all sorts of neurological conditions.
Okay, EVs are showing huge potential.
Yeah.
Now let's dive into this nose-to-brain route. Why the nose? It seems a little strange for treating brain diseases, doesn't it?
Well, as we talked about, traditional methods usually involve going through the bloodstream, and that blood-brain barrier makes it really hard to get enough of a drug or therapy into the brain. So over the last decade or so, there's been this growing interest in exploring a more direct route from the nose to the brain, particularly using the olfactory region.
The olfactory region. That's the area responsible for our sense of smell, right?
Exactly. The thinking is that intranasal delivery could let us bypass the bloodstream and that pesky blood-brain barrier altogether. We could potentially deliver all sorts of things: viral vectors, proteins, peptides, stem cells, and of course our focus today, EVs directly to the brain, non-invasively.
And this isn't just theoretical, is it? What types of CNS problems are researchers actually looking at using this delivery method for?
It's a pretty wide range, actually. We're talking neurodegenerative diseases like Parkinson's, MS, Alzheimer's. We're also talking about psychiatric disorders like autism and schizophrenia, and even neurotrauma, the kind that results from things like perinatal brain injury, status epilepticus, and traumatic brain damage.
So a whole spectrum of conditions. What makes this nasal delivery so appealing from a patient's perspective?
Well, practically speaking, it has a lot going for it. It's painless, no needles or anything, it's simple, it's easy to repeat, and it seems to work faster than other methods. Plus, patients generally tolerate it very well.
So potentially fewer hospital visits and invasive procedures.
Possibly, yeah, for certain treatments. And here's another thing. By delivering the therapy through the nose, we can often get more of it to the brain because it doesn't have to go through the liver and the digestive system first. Those systems can break down drugs before they even get a chance to circulate. So with nasal delivery, we're talking about increased bioavailability in the brain.
Makes sense. You want to make sure the treatment is actually getting to where it needs to be. But there have to be some drawbacks to going through the nose, right? It can't be perfect.
You're right, there are definitely limitations. The way the nasal cavity is structured restricts how much liquid you can give at one time. Plus, the nose has its own defense mechanisms. It has enzymes like cytochrome P450, peptidases, and proteases. They're there to break down foreign substances, but they could also break down our therapeutic EVs.
So the nose has its own security system we have to get past.
Precisely. And then there's mucociliary clearance. That's the constant movement of mucus that traps and removes stuff we inhale. And efflux transporters like P-glycoprotein, they can also limit how much gets absorbed. Even simple things like allergies, nasal polyps, or just a common cold can affect how well the nasal passages absorb substances. Oh, and let's not forget the EVs themselves. Their properties, like their size and how they're prepared, can also affect how well they're absorbed. So we really need to understand how all these factors work together and affect how EVs are administered and how stable they are. This is key for making this delivery method as effective as possible.
So it's not as simple as just a quick sniff. We need to understand how the nasal environment works. Speaking of which, can you walk us through the anatomy of the nasal cavity and how this nose-to-brain transport is thought to happen?
Sure, so the nasal cavity is divided in half by the nasal septum. Each half has three main parts. The nasal vestibule, that's the area right inside your nostrils. The respiratory region, which is most of the nasal cavity, and it has these structures called turbinates that warm and humidify the air you breathe in. And lastly, the olfactory region, which is at the very top of the nasal cavity, and it's lined with the olfactory epithelium.
And that olfactory region is the key to bypassing the blood-brain barrier.
Exactly. It offers a direct route to the central nervous system. The olfactory nerve acts like a dedicated pathway, completely bypassing the blood-brain barrier's tight junctions. But there's also the respiratory region. It has a large surface area and connections with the trigeminal nerve, which leads to the brainstem. So this could be another way to reach both the front and back parts of the brain.
So it's not just one route. What are the main ideas about how EVs actually get from the nose to the brain tissue?
Well, one theory is the olfactory epithelium pathway. This involves both intracellular and extracellular transport. So EVs might get taken up by olfactory neurons through endocytosis, where the cell basically swallows the vesicle. Then the EVs could travel along the neuron's axon, go through the cribriform plate, and arrive in the olfactory bulb, the brain's smell center.
So they're hitching a ride on the smell neurons. Cool. What's the other possible way?
It's called the indirect pathway. EVs could cross the nasal epithelium, enter the blood vessels in the nasal mucosa, and get into the bloodstream. From there, they might be able to cross the blood-brain barrier or even the blood cerebrospinal fluid barriers, through things like receptor-mediated transcytosis or endocytosis, depending on the EV's properties.
So this could still reach the brain through the blood, but they're taking a shortcut through the nose. Is there any evidence for these pathways?
Oh yeah. For example, in mice with Alzheimer's, researchers used fluorescently labeled MSC-derived EVs given intranasally. They saw these EVs building up in immature olfactory sensory neurons. Then, they tracked them as they went through the cribriform plate, concentrated in the glomeruli, those are structures in the olfactory bulb, and moved to mitral cells. Finally, they spread to other brain areas.
That's pretty strong evidence for the olfactory pathway. But it sounds like we still need more research to map out all the ways EVs get to the CNS after intranasal administration and figure out what makes some pathways more dominant.
You're right. There's still a lot to learn.
Now, we've been talking about animal models, mice, and rats mostly. But how much of this applies to humans? Our noses are different.
That's an important point. We can't just assume what works in rodents will work exactly the same in humans. There are key anatomical and physiological differences.
Can you give us some examples of those differences, especially in how the nose connects to the brain?
One big difference is the size of the olfactory area. Rats, for instance, they breathe only through their noses and rely heavily on their sense of smell. Their olfactory region is about 50% of their nasal cavity. In humans, it's only about 3%. Our nasal cavity is also much larger overall. So it's possible that the olfactory pathway is more important in rodents than in humans for delivering the same stuff.
That's a significant difference. Any other important distinctions?
Yeah. Cerebrospinal fluid, or CSF renewal, is different too. In rats, CSF is replaced about every hour. In humans, it's much slower, taking around 5 hours for a complete turnover. This could affect how quickly drugs or EVs spread from the CSF into the brain after nasal administration. That slower turnover in humans might mean it takes longer for the therapy to reach the brain compared to rodents.
So we need to be careful about assuming human doses based on animal studies.
Exactly. It highlights how important it is to adjust the EV dose for each animal model, taking these anatomical and physiological differences into account. This will help us get a more accurate idea of what might happen in humans.
Right. Careful scaling is key. Now let's talk about how researchers actually see these EVs moving into the brain after nasal administration. What kind of tracking tools are they using?
One common method is using PKH lipophilic dyes. These are fluorescent molecules that insert themselves into the EV's membrane, basically labeling them. Then, researchers can use imaging techniques to see where the EVs go in the brain tissue.
So they can literally watch the EV's journey through the brain. That's neat. But you mentioned there might be a problem with these dyes.
Yeah, it's important to remember that these dyes can sometimes clump together and form their own little fluorescent nanoparticles. This could give false positives in the tracking data. Also, the dye itself might change the EV's size and surface a bit, which could affect how they're taken up by cells and spread in the body. So while these dyes are useful, we have to be careful about how we interpret the results.
So not every fluorescent blip is necessarily an intact EV. What have these studies generally shown about where the EVs go after intranasal administration?
Studies in mice using PKH26 labeled EVs have shown that they can reach the brain quickly, sometimes as fast as 30 minutes after being given nasally. The highest concentration usually appears around an hour later. While the labeled EVs show up in different brain areas, the olfactory bulb seems to be where they accumulate most, which makes sense given the direct connection. And when researchers looked closer, they found the EVs mainly inside neurons, but also in microglia and astrocytes.
And did they find any unwanted effects in other parts of the body after giving the EVs nasally?
Importantly, giving these MSC-derived EVs nasally didn't seem to cause any noticeable changes or toxic effects in the other tissues they examined. So early data suggests it's a pretty safe method.
That's reassuring. Now, what about those animal models of neurological diseases? Do the EVs just spread randomly in the brain, or do they go to the specific areas affected by the disease?
This is really interesting. In mouse models of conditions like stroke, autism, Parkinson's, Alzheimer's, and status epilepticus, intranasally given MSC-derived EVs have shown a remarkable ability to target and accumulate in the brain areas specifically affected by each disease. This targeted accumulation was seen for up to 96 hours after a single dose.
Wow, so they're not just wandering around aimlessly. What about healthy mice? Did they see the same accumulation?
Nope, quite the opposite. In healthy mice, the EVs spread out more evenly and cleared out much faster, usually within 24 hours.
So it seems like the diseased brain is sending out some kind of signal that attracts those EVs. Any ideas what that signal might be?
Well, the evidence points to inflammation being a key driver. When researchers messed with the EVs, either by removing some surface proteins or blocking chemokine receptors, they weren't as good at finding the injury site. And they also lost a lot of their therapeutic effect. So it seems those specific membrane proteins and their corresponding receptors on the EVs are crucial for this targeted delivery to areas with inflammation or damage. It's almost like the EVs have a GPS system for damaged brain areas.
So the EVs are more than just containers. They have specific mechanisms for finding their targets. All this sniffing of EVs seems to be leading to some real benefits in these animal studies. How do these outcomes compare to using more traditional injections like IV or IP?
Well, the research shows that giving MSCs, or their secreted factors, including EVs, through the nose can be just as effective as those more traditional routes, like intraperitoneal or intravenous injections. Some studies even found that MSC-derived EVs, given either nasally or intravenously, get into the brain and move to the right spots in a very similar way.
That's amazing, especially for a method that doesn't involve any needles.
It is. While intranasal delivery looks really promising, there's still a lot to figure out. We need to know more about the differences between this and other methods when it comes to the minimum effective dose, how the EVs spread throughout the whole body, what happens to them before they reach the brain, and how fast they get to the damaged areas compared to other methods.
Lots of important details to work out, but the fact that nasal delivery is non-invasive and easy to repeat is a huge plus, especially for conditions that might need long-term or frequent treatment.
Absolutely. Now let's talk about a big challenge in EV research: the lack of standardized methods. If every lab is doing things differently and reporting their data in different ways, it's hard to compare studies.
That makes sense. How big of a problem is this lack of standardization in EV research?
It's a significant problem. The different ways labs isolate EVs, measure their concentration, and characterize them make it really difficult to compare the results of different studies, especially those in animal models of neurological diseases.
So it's like everyone's speaking a slightly different language, making it hard to communicate and understand each other.
Exactly. The scientific community has tried to address this. The Minimal Information for Studies of Extracellular Vesicles 2018 guidelines, or MISEV 2018, were created to try to unify the field and set some standards for EV research.
Sounds like a much-needed step forward. What are some areas where this lack of standardization is especially troublesome?
One example is that we don't have universally accepted markers to tell apart different types of EVs, or say for sure where they came from in the parent cell. While the term exosomes is still used a lot, meaning they come from the cell's endosomal system, we don't always know for sure how many small EVs are made. This makes it difficult to distinguish them from other vesicles that might bud off directly from the cell membrane. Their protein composition and size can be very similar. So because of this uncertainty, experts now recommend using the more general term small extracellular vesicles, or just EVs, until we understand their origins better.
So even defining and classifying these vesicles is a challenge. What about the actual methods for extracting them from the cells? Are those standardized?
Not really. Many methods are used to isolate EVs. The most common ones are based on ultracentrifugation. This separates particles based on their density, size, and shape by spinning them at really high speeds. We have differential ultracentrifugation and density gradient ultracentrifugation. Differential ultracentrifugation is often considered the gold standard, but it needs a lot of starting material, and the high forces involved could damage the EVs.
That sounds complicated and potentially harmful to the EVs. Are there any other ways to isolate them?
Yes, several alternatives have been developed. These include size exclusion chromatography, precipitation methods, filtration techniques, microfluidic devices, and immunoaffinity-based methods. But even with these alternatives, different labs use different variations. For example, they might use different types of size exclusion columns or commercial isolation kits with unique ingredients. This can lead to differences in concentration, purity, size distribution, and ultimately how well the isolated vesicles work.
So even the first step of getting the EVs can introduce variability. What about making sure the EVs are good quality? Are there standardized ways to check that?
Usually a mix of physical and biochemical analyses is used. Physical analysis looks at things like shape, size, and concentration of the vesicles, often using light scattering techniques like nanoparticle tracking analysis, NTA, or dynamic light scattering, DLS. Transmission electron microscopy, TEM, is also used to see the vesicles and make sure they're intact. Biochemical analysis looks for known EV protein markers using techniques like Western blotting, flow cytometry, mass spectrometry, or microfluidic assays. It's also crucial to include negative controls in these analyses to check for contamination. The problem is that even when you apply different isolation methods to the same cell culture sample, you can get EVs with very different protein profiles. This makes it even harder to compare studies that use different isolation methods.
It seems like every step, from isolation to characterization, can add to the variability. What about actually counting the EVs in a sample? Is there a standard way to do that?
Quantification is another big challenge. The most common ways are measuring the total protein content or counting the total number of particles using techniques like NTA. But protein measurements can be affected by the assay used and the presence of other proteins, while the particle count doesn't tell you about the protein content inside the vesicles. So the current recommendation is to use more than one method and report the protein-to-particle ratio. This gives a more complete picture and makes it easier to compare different studies.
So more detailed and multifaceted reporting is needed. And it's not just the EVs themselves, right? The MSCs they came from can also be different depending on their source and how they were grown.
Exactly. The source of the MSCs, like whether they're from bone marrow, fat tissue, umbilical cord, and whether they were preconditioned before their EVs were collected, can also influence the EVs' characteristics and how well they work.
It really does seem like a lot of factors can make it tough to compare results from different research groups. What's the main message about standardization in EV research?
Well, the lack of good quality control and standardized protocols is making it hard to compare findings and move the field forward. To make MSC-derived EVs into real treatments for neurological disorders, we need more consistency in how they're prepared, characterized, and measured. This is super important for making sure that the great results we're seeing in the lab can be replicated and actually benefit patients.
Standardization is key. Now let's get back to the exciting stuff. How do these MSC-derived EVs actually work in the brain? What are the main ideas about how they do what they do?
The amazing therapeutic effects of EVs are largely because of their diverse cargo. They carry all sorts of bioactive molecules, cytokines, growth factors, signaling lipids, mRNAs, and miRNAs. These components can affect key processes that are often messed up in CNS disorders, things like immunomodulation, angiogenesis, and neurogenesis. And because neurological diseases are usually complex, the fact that EVs can act on multiple pathways at the same time could be a huge advantage.
So they're not just a single treatment, but a whole toolkit.
Precisely. For instance, MSC-derived EVs have been found to contain a lot of immunoregulatory cytokines and growth factors like TGF-01, IL-6, IL-10, and HGF. These molecules can help reduce excessive inflammation, which is a major cause of neuron damage in many neurological conditions.
Sounds like they could be crucial for calming down an overactive immune system in the brain. What about promoting the growth of new neurons and repairing damaged circuits?
EVs are also loaded with miRNAs, which can help with those regenerative processes. For example, miR-133b promotes neural remodeling, helping damaged connections to reorganize and potentially recover. miR-219 is involved in myelinating nerve fibers in the CNS, which is essential for proper signal transmission. miR-1000 regulates the release of neurotransmitters, and the miR-1792 cluster inhibits PTN, a protein that blocks axon growth, so they could help nerve fibers grow. It's like these miRNAs are instructions delivered to the cells telling them to start repairing and regenerating.
So miRNAs are like little switches that control how cells behave. What about the proteins EVs carry? What beneficial ones have they found?
They found a bunch of proteins involved in angiogenesis and neurogenesis in MSC-derived EVs. These include VEGF, mTOR, WNT3A, PTX3, and MMP9. These proteins stimulate new blood vessel growth, which improves oxygen and nutrient supply to the damaged brain, and promote the creation of new neurons, which is key for functional recovery. Some EVs even have enzymes like neprilysin, which can break down those harmful amyloid beta protein clumps we see in Alzheimer's, so they can both deliver good stuff and help remove bad stuff.
Multitasking at its finest.
And when they looked at the proteins, they also found a lot of antioxidant molecules in EVs, things like SOD1, TXN, PRDX6, and HSP70. These can help reduce oxidative stress and neurotoxicity, which contribute to neuron damage in many neurological diseases.
It seems like EVs are incredibly well suited for dealing with the complexities of CNS diseases. But as you mentioned, we need a better understanding of how all these molecules work to develop more targeted and effective therapies.
Absolutely. And that brings us to the exciting part. What kind of therapeutic effects have we actually seen in animal models of neurological diseases after giving these MSC-derived EVs intranasally? Let's look at some examples.
Let's start with neurodegenerative diseases, like Alzheimer's. What have preclinical studies using intranasal EV delivery shown?
Okay, so in a well-established mouse model of Alzheimer's, the APPPS1 transgenic mice, giving adipose tissue-derived MSC-EVs through the nose for 2 weeks significantly reduced amyloid beta plaque buildup, a hallmark of Alzheimer's. It also decreased microglial activation, reversed neuron shrinkage, and loss in important memory-related brain areas, and most importantly, improved memory in behavioral tests. Similar results were seen in rats. Bone marrow-derived MSC-EVs loaded with the amyloid-degrading enzyme neprilysin and given intranasally for 14 days improved learning and memory, reduced plaque buildup in the hippocampus, and protected neurons. They did this by modulating the immune system and suppressing oxidative stress. In another study, preconditioned BMM MSC-derived EVs reduced microglial activation in a different mouse model of Alzheimer's, leading to an increase in dendritic spine density, which is crucial for neuron connections.
That's really encouraging for Alzheimer's. What about Parkinson's, which involves the loss of dopamine-producing neurons?
Interestingly, giving EVs from baby teeth, yes, you heard right, baby teeth, through the nose increased the number of tyrosine hydroxylase, Th-positive cells, in both the striatum and substantia nigra of rats with Parkinson's-like symptoms. Th is an important enzyme for making dopamine. This treatment also improved the rat's gait, with benefits lasting up to 10 days after the last administration. They think it works by modulating the immune system and suppressing oxidative stress and neurotoxicity. Another study using similar EVs from baby teeth also found improvements in motor function and Th expression, probably through neuroprotective and antioxidant effects, possibly involving annexin proteins.
Baby teeth, who knew? That's a surprising source of EVs. How about ALS? Have there been any studies using intranasal EVs for that?
Yep. In a mouse model of ALS, giving mouse adipose tissue-derived MSC-EVs through the nose improved motor function, protected motor neurons in the spinal cord, and reduced inflammation in the CNS. It seems like it works by using mirin A's to suppress the activation of astrocytes, which can be harmful in ALS.
So we're seeing real potential in neurodegenerative diseases. What about neurodevelopmental disorders, things like autism spectrum disorder?
This is another area where intranasal EV delivery shows a lot of promise. In studies using mouse models of ASD, like the BTBR and SHANK3B knockout mice, repeated intranasal administration of BM-MSC-derived EVs led to improvements in core autism symptoms like social interaction and maternal behavior. They also saw reductions in repetitive behaviors and cognitive rigidity and an increase in vocalizations. These positive effects lasted for up to 6 months, suggesting long-term benefits. They believe these improvements might be due to the EVs influencing GABAergic pathways, which are important for regulating brain activity. In a mouse model of schizophrenia induced by PCP, they found that the EVs helped to normalize the number of parvalbumin-positive GABAergic interneurons and reduced glutamate levels, both of which are typically disrupted in schizophrenia.
So we're seeing potential benefits in both social behaviors and brain chemistry. What about schizophrenia specifically?
Similar promising results have been observed. In a mouse model of schizophrenia, again using PCP, intranasal BM-MSC-derived EVs improved schizophrenic-like behaviors and biochemical markers. They prevented the loss of GABAergic interneurons and reduced glutamate levels, mainly in the prefrontal cortex, which is a key area for cognitive functions that are affected in schizophrenia.
So EVs could be a game changer for these complex neurodevelopmental disorders. Now, what about neurotrauma, those cases where there's a sudden injury to the brain or spinal cord?
Intranasal EV delivery injury has shown some encouraging results in preclinical models of neurotrauma as well. For instance, in rat models of perinatal brain injury, which can occur around the time of birth, intranasally administered EVs derived from human Wharton's jelly, MSCs were able to rescue myelination, increase the number of cells in the damaged area, and improve learning. The researchers believe this is because the EVs helped the oligodendrocytes, the cells responsible for making myelin, to mature and protected neurons from damage. They also found that the EVs suppress the production and release of pro-inflammatory cytokines, which are molecules that drive inflammation.
So protecting those vulnerable developing brains, what about spinal cord injuries?
In rats with spinal cord injuries, BM-MSC-derived EVs carrying P10-CRNA were given intranasally. This helped promote the growth of new nerve cells and blocked P10 activity in the injured spinal cord, leading to functional and sensory recovery, improvement in motor function, and even restoration of urinary reflexes. This was accompanied by reduced neuroinflammation, less scarring, more axon regeneration, and new blood vessel formation. They even saw improvements in the structure and electrical activity of the spinal cord. They think the EVs work by transferring pro-angiogenic myrinase and stopping those reactive astrocytes that can be toxic.
That's incredible. Regaining function after a spinal cord injury is a huge deal. What about status epilepticus, those prolonged seizures that can cause lasting brain damage?
In a rat model of status epilepticus, researchers saw that intranasally delivered human BM-MSC-derived EVs specifically went to the injured brain areas. Another study in mice with epilepsy induced by pilocarpine showed that these EVs reduced inflammation, preserved the formation of new neurons, and improved cognitive function and memory. This was likely due to their anti-inflammatory and neuroprotective properties.
So protecting the brain from seizure-induced damage, what about traumatic brain injury, or TBI?
In a mouse model of TBI, EVs derived from human endometrial MSCs were given intranasally. This treatment reduced the decline in sensory and motor function and improved neurological recovery and movement. The researchers think this was due to the EVs carrying a mix of pro- and anti-inflammatory cytokines, promoting blood vessel remodeling, and delivering factors like VEGF, GM-CSF, and IL-6, which are involved in angiogenesis, neurogenesis, and neuroprotection.
So EVs are showing potential even in cases of severe brain injury. And lastly, what about stroke?
For ischemic stroke, there's promising research too. Intranasally delivered human ATMSC-derived EVs reduce the size of the damaged brain area, improve the integrity of the blood-brain barrier, and enhanced motor and behavioral performance in rats with permanent focal ischemia. These beneficial effects are thought to be linked to the EV's ability to promote angiogenesis, the formation of new blood vessels.
Wow, so intranasal EV delivery is showing promise in a truly impressive range of neurological conditions.
Yeah.
To wrap things up, what are the main takeaways about this approach?
The research on intranasally delivered MSC-derived EVs is really exciting. We're seeing its potential for treating all sorts of neurological disorders, from neurodegenerative diseases to neurotrauma. This is largely because these EVs can target damaged areas and have multiple beneficial effects.
It's like they're tiny but mighty.
Exactly. Compared to the MSCs they come from, EVs are smaller, less likely to cause immune reactions, and better at getting where they need to go. And delivering them through the nose is non-invasive and offers a direct route to the brain.
And we're talking about a whole range of mechanisms of action, right?
Absolutely. These EVs deliver a whole bunch of molecules that do things like protect neurons, promote blood vessel growth, modulate the immune system, regenerate cells, and clear out harmful protein buildup. They really are a multi-pronged approach.
And so far, it seems they have a good safety profile.
Yes, the preclinical data from all those studies across so many different neurological conditions is really encouraging, and there's even some promising early clinical data.
But we still need more research.
Of course. We still need to figure out the best doses, the optimal way to administer them, and exactly how they work at the molecular level. There's still a lot to uncover.
But this is definitely an area to watch. The potential of intranasally delivered MSC-derived EVs.